So this is looking at respiration and digestion question. First question then, put a tick for the boxes you believe to be true of respiration. So carbon dioxide is produced is true, light is required, that's false, occurs in both plant and animals, that's true. Water is produced, that's true, glucose is required, that's also true, okay? Then it says to label the different structures, okay? So they've shown you your trachea by here, they've shown you your bronchus, so the next bit will be bronchioles, which are these bits by here, and they've done a little diagram there, which would be the alveoli, which is even bigger. From which part is oxygen absorbed from the lungs? It's the alveoli. And then this again is just kind of fits in with the Earth's atmosphere topic, because nitrogen, oxygen, and water vapour. So that's how much air, that's what's in the air that you breathe in. So, looking then at the alveoli in a little bit more detail, why do you think the alveoli is surrounded by loads of blood vessels? It allows a lot of gas exchange to occur. Name 3 ways the alveoli are adapted: large surface area, 1 cell thick, and then moist. Then kind of putting it in order of what happens when you breathe in. So it first goes through your nose, goes down your trachea, then your bronchus, then your bronchi, then your alveoli, and then to your blood vessels. Describe what happens when you breathe in then. Your ribs move up and out, your diaphragm contracts and flattens, which makes your lungs bigger. So the volume increases, the pressure decreases, so air rushes in. Next little bit then says what happens to the amount of these gases when you breathe in. Do they increase, decrease, or stay the same? So oxygen will decrease because most of the oxygen goes into your blood to make energy. Carbon dioxide will increase because you've made energy respired, so the amount of carbon dioxide being breathed out will increase. Nitrogen is likely to stay the same. You're not going to use much of that in your body. Another kind of labelling picture. So you've got your intercostal muscles here. And then your ribs are these white bits by here. Okay, so just be careful of that. It's a question. They're your ribs. And they've done them on this side, which is a bit nice of you. Structure labelled Z would be the alveoli. And then when you breathe out, your diaphragm relaxes and becomes dome shaped, so it goes back up. As a result, pressure in the thorax increases, so the air is forced out of the lungs. Now moving on to look at your digestive system, so a nice kind of labelling. It gave you the words you needed to use. So you've got your stomach, pancreas, large intestine, small intestine. What is an enzyme? A biological catalyst. And peristalsis is the muscle movement which causes food to move through the body. Again, nice little question to put them in order. So mouth first, esophagus, stomach, small intestine, large intestine, rectum, and then anus. What am I? I link the mouth to the stomach. Food moves along me by peristalsis. That's the esophagus. Food is absorbed from me into the blood. That's the small intestine. I contain hydrochloric acid responsible for churning the food and mixing it with digestive enzymes. That's the stomach. Food is reabsorbed from me. That's the large intestine. I hold feces until it's ready to be passed out of the body. That's the rectum. So, series of questions about enzymes then. What enzyme is produced by the salivary gland? That's the amylase. Breaks down carbohydrates. What enzyme breaks down protein? Protease. What is fat broken into? Fatty acids and glycerol. What enzyme works in the stomach? Protease. What enzyme breaks down food in the small intestine? Lipase and amylase. Why is it important to break down large molecules to form nutrients? Okay. Nutrients for growth, repair, respiration, and so on. Where does digestion start? In the mouth, when you mechanically break down your food. What name is given to the structure represented by B? That's enzymes. Explain why denatured protein D is unable to catalyze and separate the 2 amino acids. Substrate will no longer fit. If you look at that there, it will no longer fit in there, so it will not break down. 2 factors that control enzyme reaction: pH and temperature. Last kind of question then, looking at Viskin tubing. Okay, I'm going to draw a little diagram on the side here of what I want you to imagine Viskin tubing to look like. Okay, so Viskin tubing is like got tiny little holes in it. Okay, you will start with starch. Okay, starch is a really large molecule. Okay, just going to label that on the side for anyone who's maybe... Starch is a really large molecule. Okay, if you then add in the enzyme, I'll just draw it going around there. If you then add in the enzyme amylase, amylase breaks down starch into glucose. Glucose is actually really tiny, okay? Really tiny. So glucose will be able to fit through the viscin tubing walls and go into the water in the test tube, okay? Whereas the starch is too big, it won't fit through, okay? So starch is too big to fit through the viscin tubing, that's what we're going to imagine with the little holes, okay? Amylase breaks down the starch into the glucose, which can move through the viscin tubing. If I was to put iodine in the viscin tubing, it would go from orange to blue, okay, in the tubing, but it wouldn't in the water because no starches fit through, okay? Whereas Benedict's would go positive in the tubing and the boiling tube because it would show a positive test for glucose. So if you just want to think that the starch is, the test is iodine, and it goes from, goes from orange to blue, and glucose is Benedict's, and it goes from blue to orange, okay? What might be better is think of it's more like a blue-black colour, okay? So if I was to test for starch, I'd only see a positive result in the viscin tubing because it's stuck in there and nothing in the water. However, if I was testing for glucose, you'll have a little bit in the viscin tubing and in the water as well.